Welcome back. In this video, I have described some special structural components and metabolic products of bacteria that are called virulence factor. How those factors help to initiate infection and cause cell damage are also discussed. The structural components like capsule, pili, flagella, lipoteichoic acid are responsible for bacterial pathogenicity. On the other hand, enzymes and toxins are the most important metabolic products that initiate bacterial pathogenesis. In this video, I will discuss about microbial toxins in details. The Latin word toxicus means poison. Poison can be purely chemical, biological, or physical in nature. The poison synthesized by any living organism is called a toxin. The toxin produced by fungus and bacteria are generally recognized as microbial toxin. The word first used by Ludwig Brieger. Here I will discuss about the bacterial toxins only. Bacterial toxins are either protein or lipopolysaccharide in nature that produce specific effects on the host. Toxigenicity is the ability of a microbe to synthesize toxin. Toxicity is the ability of an organism to cause disease by means of a preformed toxin that inhibit host cell functions or kill the host cell. The effect can be acute, subchronic, or chronic. The spreading of toxin from the site of infection to other sites via blood is called toxemia. Diseases that result from the ingestion of toxin are termed intoxication. In some species, pathogenicity is largely or entirely due to the toxigenicity. Bacterial toxins are divided into two broad groups based on their relative cellular position. Endotoxin are structural components of gram-negative bacteria. The outer membrane of gram-negative bacteria is composed of lipopolysaccharide. The active component is lipide, which is called the endotoxin. It is released only when the bacteria is killed. After antibiotic treatment that can kill antibiotic-sensitive gram-negative bacteria, released sufficient amount of endotoxin inside the body. As a result, the blood pressure drops drastically and sometimes the effect is lethal. The condition is called endotoxic shock. The outcome are always same like fever, shock, diarrhea, and sometimes internal hemorrhage or abortion. All endotoxins are pyrogenic in nature like E. coli, Proteus, Pseudomonas, Enterobacter, and Klebsiella. When administered, it binds on the surface of monocyte and macrophages that contains CD14 or TLR4-like receptor. Binding of endotoxin trigger the release of IL-1, IL-6, IL-8, TNF-alpha from the host cell. The pro-inflammatory cytokines are endogenous pyrogens that increase the body temperature. At low dose, it induces immunity against it, but at high dose, it is lethal. That's why it is necessary to check the presence of endotoxin in pharmaceutical products like cyanocobalamin injection. The method by which the presence of endotoxin is detected is called the Limulus amoebocyte lysate. This method is based on the finding that lysate from the horseshoe crab Limulus polyphemus result in jellification, precipitation, or turbidity if endotoxin is present. It is very sensitive method and can be able to detect endotoxin contamination as low as this amount. On the other hand, exotoxin are metabolic product and sensitive to heat and chemical treatment as well as UV radiation. Unlike endotoxin, they are generally released into the environment. They are very powerful toxin than endotoxin. For example, 2500 to 3000 units of botulinum toxin produced by Clostridium botulinum can kill a 70 kilo human. And only 4 grams of above toxin is more than enough to kill nearly 8 billion people on this earth. Unlike endotoxin, exotoxin are very specific in terms of the target and effects. For example, botulinum toxins specifically bind only on the surface of neurons and prevent muscle contraction. On the other hand, cholera toxin binds on the gut cell surface and causes excessive release of electrolytes, which result in diarrhea. Due to extreme potency and lethality, most exotoxins are used as biological weapons. Several gram-positive and gram-negative bacteria can able to produce exotoxin. A specific bacterial pathogen may produce a single type of exotoxin or multiple exotoxin. Each exotoxin possesses a unique mechanism of action which is responsible for the elicitation of unique pathology. Unlike endotoxin, exotoxins vary in their molecular structure, biological function, mechanism of secretion, and immunological properties. The list of bacterial exotoxin is vast and increasing day by day. For this reason, proper nomenclature and classification is required. Exotoxins are named on the basis of several characteristics. More commonly, they are named on the basis of the target cell. For example, neurotoxin attacks nerve cells, hepatotoxin attacks liver cells, etc. Some exotoxins are named on the basis of associated disease like diphtheria toxin and tetanus toxin. Another commonly used naming system depends on the name of the organism which produces it. For example, botulinum toxin produced by Clostridium botulinum and Shiga toxin produced by Shigella. Now let's check how they are classified. Exotoxins fall into three main categories: the cytolytic toxins, the AB toxin, and the superantigen toxins. The cytolytic toxins disrupt plasma membrane integrity, which result in electrolytes leakage. The AB toxin consists of two subunits, A and B. The B subunits binds to the host cell surface receptor, facilitating the transfer of the A subunit across the target cytoplasmic membrane, where it damages the cell. The superantigen works by stimulating large number of immune cells, resulting in excessive inflammation and tissue damage. They are also classified according to the site of action, like neurotoxin, enterotoxin, leukotoxin, cytotoxin, and superantigen. For example, tetanus and cholera toxins are both AB toxin, but they attack different cells. Like tetanus toxin binds on the surface of neuron cells, while cholera toxin binds on the gut cell. As with other exotoxin, enterotoxin may be cytolytic toxin or AB toxin or superantigen in nature. Not only that, all cytotoxin do not target the same protein and same cellular process. Toxins normally play key roles in essential cellular processes such as signal transduction, cytoskeleton assembly, vesicular trafficking, or protein synthesis. For example, diphtheria toxin and Shiga toxin both inhibit host cell protein synthesis, but they act on elongation factor 2 and 20S rRNA respectively. Not only that, diphtheria toxin possesses ATP ribosyl transferase activity. On the other hand, Shiga toxin possesses N-glycosidase activity. Check another example. All these toxins have ADP ribosyl transferase activity, but the target are different. So it is natural that one can classify them according to the target or mode of action. You will be surprised to know that more than 400 bacterial toxins have been identified so far. Although most of the toxins produced by the microorganism are deadly, again such compounds may be used for beneficial purposes also. Most of them are enzyme in nature. Like all other enzymes, heat or chemical treatment can deactivate their catalytic properties. Some toxins, after chemical treatment, lost their toxigenicity but retain their antigenic properties, are used for disease treatment. They are called toxoids. Toxoids can be used to stimulate the development of immunity without causing disease, like diphtheria toxoid or tetanus toxoid. The toxin produced by Bacillus thuringiensis is called Bt toxin, which has insecticidal properties. It is utilized in agriculture for pest control. Botulinum toxin, also called miracle poison, sold under the brand name Botox. It is used as therapeutic agents to correct various disorders. Botox injections are noted primarily for the ability to reduce the appearance of facial wrinkles. They are also used to treat conditions such as neck spasm, excessive sweating, or overactive bladder and lazy eyes. Botox injections may also help to prevent chronic migraines. In addition, bacterial toxins have been used as a research tool to assist in defining various eukaryotic metabolic pathways like G protein mediated signal transduction. Streptokinase produced by Streptococcus, a potent plasminogen activator. It is used to clear blocked arteries in patients who have heart attacks. The information for toxin production actually encoded in bacterial plasmid or chromosome. Some deadly toxins are acquired from chromosomal material of lysogenic bacteriophages. The information integrated into the bacterial genetic material and gradually inherited. This is called lysogenic conversion. The following bacterial exotoxin are the result of lysogenic conversion by bacteriophages. So that's all for now in this introductory video. In the next video, I will talk about exotoxin in great detail. I hope you enjoyed this video, so press the subscribe button and don't forget to press the bell icon for more. Press like if you like this video and post your questions in the comment section. Visit the channel community section for more. And finally, thanks for watching.